professor at UC Merced, and she's one of the stars, I think, in the field of evolution of antibiotic resistance. I saw her give a talk in January, and it was completely mesmerizing, and so I'm incredibly thrilled to introduce Miriam, who's going to talk about predicting antibiotic resistance.
Okay, thank you so much for that nice introduction. You guys, I hope you're all mesmerized by the end of this, because you might end up feeling like you've been run over by a bulldozer. I want to show you things about how you predict evolution and using antibiotic resistance as a model system for that. And this is something I've been working on for 17 years. I am not going to give you everything I have done for the past 17 years, but I've picked and chosen the things that I think tell us really how we can predict evolution. And it does go over the course of 17 years, and it's kind of, anyway, I'm like, oh, is this going to be too much? So I'm going to be kind of hitting things at kind of the detail level where I want you to understand what's going on. But if you have deeper questions, feel free to ask. And hopefully this is just kind of a fun overview of ways of doing this. Okay. Antibiotic resistance is an awesome model for understanding evolution because it has evolved recently in response to known selective pressures. It has evolved rapidly. It is evolving in real time where we can track exactly what's going on. And the reason for this is that antibiotics were introduced. Well, there were sort of antibiotics back in the 1930s, but they weren't very good. Penicillin came out in World War II. It changed the outcome of World War II because all the wounded soldiers were able to go out and fight again another day instead of losing limbs from infections. And it has changed the whole history of the earth. Up on the top of this timeline is deployment of antibiotics. Down here is when resistance to those antibiotics is first observed. In every single case that has been documented, resistance is usually showing up within 3 years of the deployment of an antibiotic. Now there are some here, if you're really careful, you might see that there are lengthy delays for some, like for vancomycin, and those are because a drug was invented and then it wasn't used very much. And so it just, yeah, and then as soon as it starts being used any appreciable amount, boom, there's resistance overnight. Okay, and so it's this amazing model because this has happened over and over and over so many times, and we have a record of all of it. The resistance I'm going to focus on the very most is resistance to beta-lactam antibiotics. Penicillin is a beta-lactam antibiotic. Here is penicillin. Beta-lactams are characterized by this four-membered beta-lactam ring. It interferes with cell wall synthesis. There are many varieties of beta-lactams. There are inhibitors, clavulanic acid is one of those, carbapenems. These are kind of the things you're hearing about the most if you've heard about CREs in the news at all. Those are usually E. coli or Klebsiella that are resistant to this kind of drugs. This is where resistance is emerging right now. And then there are monobactams, which are a lot like cephalosporins, and there are cephalosporins. These have been the most heavily used antibiotics since the 1940s when they were introduced. And so because of their heavy usage, there's been a lot of resistance that's evolved to them and a lot of different resistance genes. And so this is kind of the foundation of an amazing model. It may also be the foundation of the end of the world, but it's an amazing model to learn about in the meantime. Okay, so the thing that causes resistance to beta-lactams usually is a beta-lactamase. This is the 10-beta-lactamase in particular. There's a beta-lactam sitting in the binding pocket. Entry is kind of through here into it. There's this omega loop in many of them that it kind of regulates what can get into the binding pocket, and so a lot of the mutations that have resulted in resistance occur in this omega loop. But there are mutations throughout the entire enzyme that have done this. Most of them destabilize the enzyme or destabilize the omega loop a little bit and make it a little bit floppier. Okay, let's see. Okay, so let's look at different ways of predicting. So the first thing I did in trying to predict how evolution is going to happen is I made a whole lot of phylogenies, and I'm going to show you a bunch of phylogenies, and we're just going to kind of hit the highlights of them because phylogenies make people yawn a lot. And anyway, I'm starting out with a cichlid phylogeny, which is, I don't study cichlids, but cichlids I think are a good example of kind of what I'm going to be talking to you about. So are you guys familiar with cichlids? Okay, so you put some ancestral cichlid in a lake, and it can, there's some argument about this, but I think it's pretty well established that they can adapt many, many different niches within this lake, and they come up with a lot of weird different forms they can be in, and it's super cool. And so if you, okay, and so then you can compare them, say, to a pufferfish, which, what does it do? It does this one really cool thing, and that's it. And it's not, you're not getting hundreds of different varieties of this pufferfish. And so if you're gonna like try to adapt things to a new environment, which would you bet could do it? A cichlid or a pufferfish? Probably a cichlid. We've seen them evolve many different morphs over and over and over again. And so that's probably a good indication that they can do this again, even if we expose them to possibly new environmental conditions, right? And so that's kind of the story that's going on with beta-lactamases. Here is a beta-lactamase phylogeny. Okay, beta-lactamases are really, really old, like origin of life old almost. They go clear back before the split between gram negatives and gram positives. They, you know, they're on the order of like 3 billion years old, or so. I mean, we can't put really clear dates on them going back that far. They're really old. There are 3 different classes of them which separated way before the gram negative, gram positive split. There are the class A's, the class D's, and the class C's. The thing I want you to, okay, these are just, you know, very select taxa, but we tried to represent kind of the major, well, kind of represent what's going on in these, at least enough to get a good sampling of the phylogeny. And the first thing I want you to notice is that there's more going on in the class A group. The class A group is a lot bigger than the other groups. And yeah, this has been edited, but it is a lot bigger than the other groups. And if we were to go and pick every single known one, this would still be the biggest by a long shot. It would be bigger by even a larger margin than it is here. And so that already gives us some indication that the class A's may be a little bit more evolvable. Here is a class A phylogeny. Okay, and within this phylogeny, there are groups that are really big. Here's a CTXM group. I'm going to use laser pointer. These I had to prune out totally, the TEM and SHV groups. It's because each one of these groups has many hundreds, and so we just put in 1 to represent the whole group. But you end up finding these groups in here where if you check for positive selection, there's a ton of positive selection. There's been a ton of evolution in these groups. And then, I mean, we've gone through and we've done very specific little trees of alleles of many, many of these other genes in here, and you don't tend to see positive selection so much for them. You don't tend to see that many alleles or that many variants in it, and you also tend to not see them having much clinical impact. And so it seems like these groups where there is a lot of positive selection, they're also the ones making a lot of clinical impact. They're also the ones where there are many members of them. And we're not gonna look at the SHVs because their story is almost identical to the TEMs, but we're gonna look a lot more closely at the TEMs. Okay, if we go to the class C phylogeny, okay, this is a little bit edited, but it's not that edited. There are some groups that have experienced some positive selection, and this tree is a little old now. We could probably double or triple the size of some of these groups at this point, but they're, you know, this is not a very much edited tree, and this is kind of what we've got. There's some evolution going on. These are important. Class C's are the AMP resistance that you see in E. coli. They're in the genomes of many different organisms, and they are somewhat evolvable, but they have not gone quite as crazy as the class A's. Another is the class D's, and these We have some where there's been a lot of positive selection, a lot of recent evolution, but again, even less here than the class Cs. And there is some positive selection, but not as much as what you would see with the class As. Okay, so let's zoom in on some of the class A phylogenies. Here's the CTXM group. These started showing up in 1983, actually in a dog. The first one was found. These are rapidly becoming the most abundant resistance genes in the world. There are more than are shown here in this tree. There are like more every single day, and kind of getting to be so many that you can't put them all in one tree that anybody can look at. They've had a huge amount of positive selection. So CTXM3 is an ancestor here, and we can track what organism it came out of and look at its evolution. And so these have been a very evolvable group. The other group that has been super evolvable is the TEMs. We're going to be looking at the TEMs more closely. They have 3 main phenotypes that they've been able to evolve. These guys are acquiring, if you try to date this tree, so back here is 1963, and if you go out to the tips of the tree, they are accumulating about 1 mutation a year. This tree is a little, you're like 1 mutation a year, I don't see it here. This tree is very edited because there would be about 500 in this tree, 500 taxa in this tree if we didn't edit it. And so if you're looking at that, they acquire about a mutation a year. So they are zooming along evolutionarily, tons of positive selection. It's kind of hard almost to quantify how much positive selection because nearly every mutation is an amino acid substitution, and it's just like entirely positive selection. Okay, and there are these 3 phenotypes. There's parental, which is resistance to penicillin. These guys emerged to be able to confer resistance to penicillins. They have evolved the ability to confer resistance to cephalosporin. So a lot of these out here confer cephalosporin resistance. And some of these are resistant to inhibitors. So you may be familiar with some inhibitor penicillin combinations. Augmentin, I don't know if your kids get ear infections and the doctor gives them Augmentin. But it's penicillin plus an inhibitor, or amoxicillin plus an inhibitor. And so, anyway, so there are resistance in this group to all 3 types of antibiotics. And we're going to look more closely at these. The conclusion we can make from these phylogenies is that some resistance genes have more evolutionary potential than others. And when you're looking at where new resistance phenotypes are going to come from, which I've been doing for quite a while now, it always seems to come from the groups where resistance has come before. Does that basically, I mean, that's a lot of phylogenies for this little tiny point, but that's where it seems to come from. And I've been watching and I'm like, can we get a new one to emerge? Not that that's a good thing, but I kind of get excited, but in, you know, kind of grim way. Anyway, and it usually doesn't happen. Occasionally there will be a new one that emerges. CTXM's emerging was a little bit of a surprise, but for the most part, it's the genes we've already got and know about that are evolving the resistance. Okay, so that gives us some basis for predicting who the major players are going to be. How are they going to play this game? How are they going to evolve? What mutations are going to arise in them that are going to cause the evolution of antibiotic resistance? Okay, so we came up with this method, which my advisor calls the Barlow-Hall method. I'm like, oh, Barry. Anyway, I don't put that up there. Anyway, so what it is, it's an experimental evolution method. You take your resistance gene and you amplify it by PCR and you use a mutagenic PCR method. So you just mutate this gene a lot. You don't want to mutate it so much that you have many mutations in every single copy of the gene, but what you would like is 1 to 2 mutations in about every copy of the gene. And and you have some with 3 and some with 4, and it's this Poisson distributed thing. And then you take these copies that have these mutations and you clone them and you create a library and you have to make really big libraries. I spent a lot of years making really big libraries, and it's devastating making really big libraries, because it's like, okay, one wasn't enough, let's go make 50 more and see if that gets us there. And it seems like the closer you get to 50, the smaller they get with every iteration. Anyway, you make all these libraries, hundreds and hundreds of libraries, and anyway, you get them to a large enough library where you can sample all of the single and at least 95% of the double mutations, and then and then it's like, well, you're going to be doing this forever with no publication, so at some point you just stop and work with what you've got. Okay, so you take these and you amplify this library, and then you put it through different selective conditions. So here, what this is showing is one antibiotic at 3 different concentrations. And what you want to see is it growing at a higher concentration than your original. You want to see these enabling bacteria to grow at a higher concentration of antibiotic than these guys could. And then you look at what the mutations are, and you say, okay, these are the mutations that are going to result in resistance to this drug. Now, when you're doing this, you have to be careful, because you've got a gene that already exists, and it's doing its thing. And if you only select for it to do a new thing, this may not be what's happening in nature, because it's still got to do its old thing, right? You're looking confused. Anyway, so if we look at the B-lactamase, which is the gene that I've done this to a bunch of genes now, but we're going to look at TEM data first. So this is all for the B-lactamase. So you take TEM1. It's good at hydrolyzing penicillins. And we want to say, can we reproduce its evolution of cephalosporin resistance? Okay, so we go and we cycle it through a bunch of cephalosporins. This is a monobactam, but that's a lot like a cephalosporin. And then we go and we passage it through other drugs. Penicillin, that it caused resistance to, a different kind of penicillin that's a little bit more modern, that is conferring selective pressure, and then cephalothrin, which is a really old cephalosporin that TEM1 actually can hydrolyze. So we're selecting for a new function by cycling through these drugs, and then selecting for retention of this old function. Okay, and so, well, phenotypically this worked. We took TEM1. So here, okay, up here penicillins, down here are cephalosporins. TEM1 is not very good at hydrolyzing cephalosporins. Down here it's resistant at 16 micrograms per milliliter, or 0.25 micrograms per milliliter, or 0.125 micrograms per milliliter. So this is pretty low resistance, and its ability to survive in increasing concentrations of antibiotics is shown here. So we've moved it from being able to survive in like 0.25 micrograms per milliliter of cefuroxime up to 512 micrograms per milliliter of cefuroxime. Are those data clear? You can understand what's going on with that. So it's become more resistant to these drugs. Okay, and so then we look at the mutations that we recovered in doing this. Now we had identified 9 mutations that have occurred independently multiple times over the course of TEM evolution. And the ones that are shown in parentheses here are those that have occurred independently multiple times in nature. So these are the important ones that have been important for this function of evolving cephalosporin resistance. And so we captured 7 of the 9 of these, and we went, that's pretty good for in vitro evolution. We'll take 7 out of 9 as success. We can reproduce the evolution that has already happened. And so then, of course, what do? Then we use this method to predict how evolution is going to occur for new antibiotics. Now, at this point, this is a difficult question to address because there are no new antibiotics. There are a few new ones coming out, but in 2005, Big Pharma bailed on making new antibiotics. But back in the day when I was doing this, there was a new antibiotic. It was named cefepime. And so I wanted to see if TEM could evolve the ability to hydrolyze cefepime or confer resistance to cefepime. So we start out with, and so here, okay, here's the TEM1, the concentration of cefepime where TEM1 can grow. And we evolve it, and we indeed see that we can get a higher concentration. And then we look at what mutations are there. And there was a new mutation that no one had ever seen before, I173V. So this is at amino acid position 173. We change from an isoleucine to a valine, and boom, we get cefepime resistance with that change. And this was something no one had ever seen before. Shortly after we published it, this mutation was found in a clinical isolate, and we went kind of boo, but kind of yay, we were right. It's nice to be right, even if it means things are getting worse in the world. It's going to happen anyway. Let's predict what's coming. I don't know. Anyway. Okay, so it worked for TEM. It didn't work for CMY2 though. So this is an AMP-C that it's on a plasmid. It causes a lot of troubles. It spreads around. Okay, so CMY2 can grow in 2 micrograms per milliliter of cefepime, and we did get higher resistance, 64 micrograms per milliliter it could grow in. The level required for clinical resistance is 32 micrograms per milliliter. So we're right around, just barely the threshold for clinical resistance, which is not overwhelming. We got higher levels out of the TEM, but anyway. And so this, it's like, okay, this could do it, but maybe it doesn't do it very well. Here are the mutations that we found. There are a few that occurred multiple times in different lines, A292P, L293P. Anyway, we had a few of these, but none of them, to my knowledge, have ever been found in clinical isolates. So we go for CMY2. This just, we made predictions about what was going to happen, but it didn't work. This is a class C beta-lactamase. They're not as evolvable. The resistance didn't go as high. This didn't end up being the solution that nature chose. And I think that's a little bit important, emphasizes a little bit of the importance of why we need to look at phylogenies too, because I think from phylogenies you find the most likely candidates. You can verify what's going to happen in the lab by trying evolution there, but anyway, this was a ton of work, and it's just like, okay, we got these predictions. I'm still waiting. They're never going to come true. I'm okay with it. I've made my peace with this. We tried this out on CTXM. Okay, so CTXM ends up being the real solution that nature chose for cefepime resistance. It's this gene. It was already out there before cefepime was introduced, but you introduce cefepime into widespread clinical usage, and this gene spread all over the world super quickly. It's still spreading. It's still displacing TEM in bacterial populations, which is this whole thing that it's very interesting that it can actually displace other resistance genes, because everybody's like, well, bacteria can just acquire zillions. No, that's not what they do. This is displacing TEM. And so it's really good at conferring resistance to cefepime. Greater than 32 just means it can grow way above the clinical resistance. But then, okay, it's not very good at hydrolyzing ceftazidime. So I worked with Carrie Welsh, who was a master's student, and we evolved this to be able to hydrolyze ceftazidime, which it evolved the ability to do, but in doing so lost the ability to hydrolyze cefepime. So there were definite trade-offs in this. We found this one mutation that was very responsible for that function, which has since appeared in natural populations, and this is a solution that nature has chosen. And so I present this idea of doing in vitro evolution as a way of predicting evolution to you, to show you that, this really can work. It can predict what's going to happen in the real world, in the future, but not all of the predictions are necessarily going to come true. You have to know stuff about like how evolvable these genes are. Are there genes that can do a better job than this, that bacteria are going to like better than this gene I'm working on? So there are multiple factors to take into account here, but it's an interesting thing to think about if you want to predict how evolution is going to happen. Okay. So conclusion, some genes have more evolutionary potential than others. We've seen it in phylogenies, we have seen it in experimental data, and new genes can emerge to cause resistance. Okay, so we can predict how things are going to happen. Can we make these predictions actually be useful predictions? And so if we're going to make these predictions, we would like to maybe make the world a better place instead of just cheering for the enemy every time they have a success. And so we ask the question, how is antibiotic consumption going to affect the evolution of antibiotic resistance? And I've already shown you, like as for CTXMs, as they evolve ceftazidime resistance, they lose cefepime resistance. If you're not selecting for the retention of penicillin resistance in TEM, you're going to lose that. But even though you hang on to it, actually evolving cephalosporin resistance cuts it just a little tiny bit. They get a little less good at hydrolyzing penicillin. And so we wanted to use these trade-offs in resistance to see if we can come up with better ways of using antibiotics. So we've done this based on in vitro data, but this work is computational work that I'm going to show you. We haven't tested this out in the hospital, but it's still fun. Okay, so this is the paper we published, Rational Design of Antibiotic Treatment Plans, a Treatment Strategy for Managing Evolution and Reversing Resistance. Okay, what we did in this paper. There is this TEM gene called TEM50, and it has 4 mutations in it. This is a really exciting gene because it's devastatingly horrible. These 2 mutations make it resistant to beta-lactamase inhibitors. These 2 mutations make it cephalosporin resistant. Now, these kinds of mutations do not get along, the inhibitor resistant and the cephalosporin resistance mutations. The cephalosporin resistant mutations, it's because they're making this omega loop floppier. They're opening up the binding pocket so that the cephalosporin can fit into the beta-lactamase. These, on the other hand, are blocking the inhibitor from binding into the active site. And so on one hand, you've got things that are opening the binding site up, and on the other hand, you have things that are making the binding site a little more protected. They don't get along. And so this guy found a combination of 4 that get along very well. And so it can just be resistant to all sorts of beta-lactams. Still not carbapenems, but everything else. Everything but carbapenems this guy causes resistance to. So it's pretty nasty. Okay, so we wanted to study these 4 mutations and look at what the trade-offs are as it acquires all 4 of these mutations, and then see if we could find ways of reversing the evolution of this. Let's say you have TEM50 that has arisen in your hospital. Can you get rid of it? We already know from so many studies that just stopping the usage of antibiotics doesn't do it. Then you're waiting for like drift to happen in like a nearly infinite population of bacteria. You're going to be waiting a long time. And so we're trying to use selection to reverse things because selection moves really quickly in large populations. Okay. So we went and we figured out the adaptive landscape for all variants. Okay, let me go back for 1 second. So we create all 16 variants of these 4 mutations, every single, every double, every triple, and then TEM50, yeah? And then we measure their fitness in different antibiotics, each of the variants. And we present them in these landscapes, which, okay, so we've got fitness going up the y-axis, and then down here we have got the different variants. The axes aren't big enough to show all 16 of them. It's not super important. What is important is that these landscapes for different drugs are different from each other. So we do this for 15 different drugs. Okay, and then we plug them all into a mathematical model, which we call time machine. I'm not going to show you equations. No one wants to look at equations. But for those of you who are interested in math, what this comes down to is taking the fitness of all of these different guys and figuring out transition probabilities, like how likely are you to go from this to this state if we're using this drug. So we have all these transition probabilities. We take all of these probabilities for all these different drugs, all these different transitions, and we do matrix multiplication, and we multiply everything by everything, and we come up with an even larger everything, which is all possible things that can happen as we're moving through these adaptive landscapes and changing drugs. Every possible combination of drugs, every possible change, and its probability. Okay, so it's way too much to look at. I know, because when the program got written, they send me the output and I print it out, and it's this stack of papers. And so then I drove to my collaborator who wrote the program, Bernd Sturmfels. He's at Berkeley, and I drive there with this stack of papers because the phone was not going to work in this case. And I go, Baron, let me show you the results of this and what they look like. And we start going through, you know, the first 10 or 12 pages that I've gone through, and he goes, oh yeah, this isn't going to work. We need to make this better. And so then he wrote an algorithm that goes through and searches for the optimum pathways so that it's not me searching for the optimum pathways. I love good collaborators. And so we have this matrix multiplication and then an algorithm to search for the pathways that give us the highest probability of doing whatever we tell it to do. But in this case, we're telling it that we want to go from TEM50 to TEM1. So down here is TEM1, 0 mutations. Here are all the single mutants, the double mutants, the triple mutants, TEM50. And we end up finding a few pathways where you can go from here down to here, but this ends up being the most likely, and these are the drugs that can get it done, and it's with a probability of 0.617. This is not a sure bet that you're going to go from here to here if you rotate through these drugs, but this is a lot better than random. It's a lot better than making things worse, and so not perfect, but it's something that can happen, and we have a probability of how likely it is going to be. Okay, so we can add to this that we can use predictions for clinical purposes. Now, you still are trying to, well, I'm going to get into this. Everybody loved this. It was devastating how many people love this. I'm off in Pittsburgh judging a science fair, and my phone is ringing off the hook because people want to talk to me, and I'm like out on the street corner with the wind blowing, and people, oh my gosh, and trying to talk to like Scientific American and all, and I'm like, okay, whatever. And then I get back, and like everybody's mad at me because they weren't included in the press, and I'm like, I don't know, I'm sorry. Anyway, this is what happens with press. Everybody loved it. It was really good. Everyone was super happy, but they were like, where's your picture in the paper? Not my, anyway, sorry everybody. Everybody deserved to have their picture in the paper. Anyway, here's what, all around the world, and they keep asking me the same question that is super hard, and that's, we want to do this. How do we do this? And that gets me thinking, okay, how do we do this? If we're going to rotate drugs, how are we going to rotate them? If we're going to alter evolution, if we're going to use our predictions to change evolution in the real world, this is going to be hard. What is the scale that we have to do this at? We got individual patients with bacteria growing in their bodies. Okay, here, I have slides for this. We're going to go through the slides. I'll do it this way. I get too excited sometimes. So what's the appropriate scale? There is the whole world. We're going to just stick with the world. I don't think that antibiotics are being launched into the universe on spaceships at this point. Who knows? But I don't think so. Okay, it could be done at a national level. And there are arguments for each one of these. So at a global level, you know, we're getting resistance over from Japan. I love Japan. But for a while, and it's stopped now, for a while doctors were getting paid royalties or commissions based on the antibiotics they prescribed. So they're like, oh, there's a new expensive antibiotic. We're giving everybody that so we can get rich. Totally understandable. That got changed though as resistance emerged in Japan and started spreading all over the world. I mean, this is a global issue. Okay. National. Okay. People have tried things at a national level in Europe. The UK cut back antibiotic prescription for what was it, 3 years? Over that time, resistance to a couple antibiotics went down a little bit, but for other antibiotics where they really restricted usage on them, resistance was increasing over this time. And it's like, yeah, cutting back nationally didn't work there. There are other arguments, though, that it might work, maybe. I mean, England's kind of, the UK's small-ish, I don't know. Canada claims that they have much less resistance than the US. If you are a US citizen and you're going to get hospitalized in Canada, at least at one point they used to quarantine US citizens if they were hospitalized in Canada to screen them for resistance, because they did not want Yankees bringing over resistance and spreading it through their hospitals. Okay, so, I mean, Canada probably does have less resistance. Okay, at a state level, well, who knows? At a state level, though, arguments could be made that it's easy to coordinate policies, easier to coordinate policies. You may have regional things like agriculture going on that the state should be looking at as a whole. You know, each state may have separate issues that should be looked at. There's a county level. This is Merced County. County is even easier to regulate. I think there's a strong case to be made for that things could be regulated at a community level. I mean, at a community level, certainly things are going to happen more quickly there. You've got your environment, whatever it is. In the Central Valley, we have a whole lot of antibiotics being dumped into everything because there's so much agriculture all around. We also have, you know, you're interacting with these people, you're sharing germs with people, by which you're sharing resistance with people. And so maybe at a community level makes a lot of sense. Hospitals are the case where the strongest argument has been made simply because you can actually do something at the hospital level. This has been tried a bunch of times where people have tried to decrease resistance in a hospital. It hasn't worked super well, but it doesn't mean that it might not ever work. It might just be because they didn't know what they were doing. They were just randomly changing between antibiotics and waiting for drift to decrease the levels of resistance, or waiting for supposed fitness costs for carrying resistance to decrease the prevalence of resistance. Most resistance genes do not have significant fitness costs. I know that sounds like really weird, but they don't. Bacteria can carry them around quite happily when there are no antibiotics present, usually. And so, anyway, so maybe this could be done better in hospitals in a way that could work. And I think a very strong case can be done for doing this in humans. Not necessarily for every infection or every antibiotic, but like HIV patients are already having drugs cycled through them. People with cystic fibrosis also, I mean, people with chronic infections are the ones where it might make the most sense to be cycling antibiotics through them, because if you're on a lifetime course of antibiotics or antivirals, maybe having a systematic way of cycling them where you actually can predict what's going to happen as you cycle them could be useful. Okay, so to answer this question, we looked at it at a hospital level. This is just the beginnings of an answer. Why did we look at it at a hospital level? Because we're collaborating with a hospital and we have data from this hospital, which is super nice. It's so hard to get data from hospitals. Mercy Medical Center has been such a great collaborator and so sharing with us. So we just wanted to ask the very scary question, is resistance evolving in your hospital? They don't even care that we're asking this question. They're happy for us to ask this question. They have some serious courage to let us just venture into this stuff, and kudos to them, and they actually come out looking pretty well in the end. Okay, so here we have this model. This is our model for testing this. Okay, we have infected patients that go to the hospital. They have some sort of resistance thing, and then we have uninfected patients that go to the hospital for some other reason, and they, within the hospital, may end up getting exposed to this resistant strain from another infected patient. This is sort of what people think happens. You go to the hospital because you have cut your arm off, and in the hospital, the wound where you cut your arm off gets infected with a horrible strain, and then you end up, I don't know, in worse shape than when you went in. Public service announcement, don't go to the hospital unless you have to go to the hospital. Like, leave as soon as you can. It's not a place to get better. It's a place to be on life support with close access to very well-trained medical staff. Once you don't need that anymore, go home. Heal at home. Your house may look like a disaster. It's cleaner than a hospital. Gonna almost guarantee that. I haven't seen all your houses, but we're gonna go with that as the general case. Okay. So we have these individuals, they get colonized. There are bacterial isolates that get collected by the hospital. These are the kind of data we're getting. We get the isolates. We're not going to look at the actual isolates in this. We're doing other stuff with them that's cool. And then by Vitek 2, they determine if they're an ESBL. What is an ESBL? It's an extended spectrum beta-lactamase bacteria. So it's resistant to cephalosporins. That's really what that means. Resistance to cephalosporins. And then we do MIC testing or resistance testing to see exactly how resistant they are to these different drugs. Then we have this method, which my math colleagues, Bernd Sternfels and Anna Siegel, named NERD because they are nerds. And if this ever gets used in hospitals, they can be the little omniscient nerds watching over you, which is sort of comforting. Anyway, so we name it NERD. And what this is a summation of transmission probabilities as a function of our transmission model. We'll get into what that means a little more, not too heavy. Okay, and so then we come up with this probability and we compare these probabilities based on random probabilities that are derived from the overall frequencies of the resistance phenotype. So if things were just happening randomly, what would this look like? And is what is going on different from randomness? Okay, and then we do a permutation test of a Bonferroni correction. I hate Bonferroni corrections, but we do them because we're supposed to do them. If you've ever done a Bonferroni correction, you know why I hate them, but we do them anyway. Okay. So here we have randomness and here we have non-randomness. This is actually for financial stuff. It was the best picture I could find of random versus non-random though. And so we're looking for non-random patterns in the hospital. We're looking for connections within the hospital that suggest the resistance is evolving in the hospital. If resistance is evolving in the hospital, what this is implying is that a strain is leaving some infected patient. Maybe it's being carried around in the ducts, maybe it's being carried around by healthcare workers on stethoscopes, on pens, on whatever. It's somehow there in the hospital and it's going to get transmitted to another patient and then another patient and another patient. This does happen. We certainly have anecdotal cases. An anecdote, I don't mean funny. These are not funny cases. In the NIH hospital in Bethesda, they had a strain come in that they were able to track, and they still, to my knowledge, haven't been able to get rid of it. And it comes out every once in a while and infects a patient and kills them because there's pretty much nothing left to treat this strain. So that can be tracked very well. You have these things called clonal outbreaks that happen in hospitals, where somehow one clone is being disseminated through the hospital. And so based on these very clear examples of this happening, everybody kind of assumes that this is the general case in hospitals, that resistance evolves in hospitals. We know it does sometimes. Does it do it all the time? Okay, so to answer this question, we had to ask, how long does transmission take? If we're looking at a strain getting transmitted from one patient to another, that is the basis of our model, transmission between patients. What does this look like? Okay. And so we went and extracted data from all of these clonal outbreaks, hospital size, number of patients in outbreaks, and then average time from patient 0 to the next infection in days, because you can track that pretty well. You have patient 0 that has appeared, and then the next patient who ends up with this infection. It's easy to track because you got 2 people with this infection, and that's it. Okay, and so it ends up being this range. Can I just suggest to you also, don't get sick in Italy. I love Italy. I want to go to Italy. Don't get sick in Italy because it's, we'll just not even go there. Don't do it though. Anyway, So our hospital is down around in kind of this size. We ended up going with the number 100, possibly for convenience. The model that we use ends up, it's because our hospital was close to this size, but this is a NICU, which is special. But we ended up choosing 100. It's within the realms of what is reasonable for here. There is a big difference in how the model performs between 100 and 340 days, or 1,000 days, or 500 days. But there's not a whole lot of difference between 100 and 120 days, or 130 days. And so we just went with 100. Mathematicians are not scared of numbers that are not even, but if they want to go with 100, man, I'm just going to let them go with 100. I work with good mathematicians. I trust them. Anyway, we use the negative binomial distribution. I don't know what to say about the negative binomial distribution. It is a pain to work with. But what we ended up doing is figuring out, using the negative binomial distribution, the probability of any, so we look at all possible pairs of resistant strains collected from the hospital and the time that is separating the collection, the isolation of those. And using this number of 100 as the peak, most probable time for transmission, then we sum the probability of these strains being transmitted from each other, or from one to the other, just based on how far apart they are. That probably doesn't make any sense. I feel like moving on anyway, because it could make less and less sense if I keep talking about it. But if you really want me to show you on a piece of paper afterwards, and I can take some significant time, I'll do it. I can, but it takes a minute. Okay, so we do this, and we end up finding that there are 3 significant drugs that are non-random. So we do this, we sum all these probabilities, and then we do a permutation test to look at to get the significance of these, and we're comparing them to how significantly different they are from randomness. And we get 3 that are significantly different from randomness. And they are ceftazidime, cefepime, and gentamicin. Okay, and these are not Bonferroni corrected. And these 2 are not significant after Bonferroni correction, because what that looks like is taking these values and multiplying them by 16. Worst statistical thing to happen in the whole world, but we do it. This one remains significant even after Bonferroni correction. Okay, so then we want to say, okay, so we've got these 3 that are sort of significant to really, really significant. And we ask, well, what is the trend? Okay, this is the trend. It's kind of horrible to look at. But what we end up seeing is that in the recent past at Merced Medical Center, resistance to ceftazidime and cefepime are decreasing. The other thing we see out of these is that these 2 antibiotics are tracking along with very similar patterns, which suggests a single resistance determinant causing both of these trends. Now, I have told you a lot in the course of this talk, particularly about CTXMs, which confer cefepime resistance, but they're not particularly good at ceftazidime resistance. There's one exception to this, which is CTXM 15. It does cefepime really well, it does ceftazidime really well. And so we come up with this hypothesis that CTXM15 should be the basis of this trend. Gentamicin, I have no idea what's going on with it, but it's significantly different than randomness. It seems like it's this periodic thing. And we're actually looking into that more, because it seems like resistance levels may have seasonal variance. And so we want to see if that's what's happening with gentamicin resistance. If it is, I would bet you it's because of the agricultural community that the hospital serves. But I'm not certain of that. This is still like me telling you what I think, not what I know. But I'll tell you what, I can be irresponsible a little bit. So there's that. So, but then with these, okay, there's this clear trend. We think CTXM15 is causing it. Okay, so here's CTXM15 in all its glory. It is heavily distributed throughout the globe. Like, this isn't me just like coming up with random stuff. Here's CTXM15 in this kind of purplish brown color. And it is all around the globe. It is in the US as the most prevalent one, but it also does this thing of conferring cefepime and ceftazidime resistance. So We sequenced from the hospital the whole genomes of 48 isolates. We got another 125 we're working on, and we'll see if this trend holds. But out of those strains, 29 of the 48 include the CTX15 gene. And so we kind of went, woohoo! We made this model, we came up with this result, we made a prediction from this result, and our prediction came true. Therefore, our model at least has some clinical validity, though, anyway, we're curious to see how it holds up as we get more and more sequence data. But we think that it is possible to say at this point that most resistance is not evolving in hospitals, but that it can evolve in hospitals. That doesn't totally answer our whole scale question, but it's a start. This is a huge question to answer, and it's going to take a lot more than just me to answer it. It's going to take nations. Europe is kind of heading in a direction that might be informative to this, but it's going to take studies, maybe not even like this, but studies of this question at much bigger scales and smaller scales to find out what the right scale is for trying to manage the resistance problem. So, anyway. Those are my conclusions. That's my story, and I'm sticking to it. Do you guys have questions? We've still got 10 minutes. Can you believe we made it through 17 years of research in 50 minutes? Do you feel like you have been run over? Possibly. But you can ask questions. Yeah.
So in the first part, what do you think the reasons are for some genes being more evolved than others?
What do I think that it's going to be the structure of the gene. So a lot of the genes that we find are the most evolvable have this significant omega loop. And some of, like in AMP-C, it's really good at hydrolyzing cephalosporins, but it has almost no omega loop. It just has this little bloop instead of this big, nice loop. And so in doing that, it kind of makes it more of a generalist enzyme from the beginning, but I think it also makes it so that mutations to it have a greater structural effect. Like instead of loosening it up a little bit, because that's what you're doing with TEM when you're mutating it, you're loosening it up just a little bit so it can fit more things. And I think when you're doing this to AMP-C, I mean, you can still do it some, but I think it's easier probably to destabilize the whole enzyme. And so if you were going to totally change it, maybe it would require an insertion of, say, an omega loop or something like that. But I think, and so there are these major evolutionary changes that could happen that could do this, but I mean, one thing I didn't get into is that it also seems like nature is a little bit lazy. It's like the thing that arrives first. It has to be like, there can't be anything super, super better than that because it'll get displaced. But it often seems like the thing that arrives first, if it can just be fine-tuned with a mutation or two, is the thing that ends up sticking rather than bringing something in that might be, like if you have two things that are similar to each other, and maybe this one's even a little better, but this one arrives first. With a mutation or two, it becomes a whole lot better. Then this guy's going to just take over the population, and this guy is not going to take over the population. And so it seems like this, like waiting around for really huge evolutionary changes or lots of evolutionary changes is not what's happening. It's just like take the first answer, tweak it a little bit, and you call it good and run with that. Yeah.
Following up, so basically what you're saying is it has really nothing much to do with selection, but rather it's a coincidental thing about how some proteins are more robust to change than others.
Well, I mean, there is a component of selection that is involved with that, but there is also this aspect of, I mean, there's selection on the organism for which genes are good, but as far as which enzymes are going to do it, it does seem like the quick solution is the one that's going to get there, and that to take something that's less evolvable, it's going to be longer. I mean, sure, you can turn any sequence into any other sequence with enough mutations and enough insertions and deletions, but that's going to take a long time. And so, yeah, I think it's this time thing and this just what can adapt the most quickly thing. I don't know. Does that answer your question? Okay. Yes?
I have a question. So in your second part, I think you showed data on the bark evolution of B-lactamase, right?
Yep.
So I noticed there are quite a few synonymous mutations, right?
There are synonymous mutations.
In a sense, like if you see some synonymous mutations occurring in multiple clones,
Oh.
you actually try to look at.
Oh. So the synonymous mutations, okay. I mean, true stories here. Some of the synonymous mutations in these experiments, I think, were probably cross-contamination at some level, like at round 1 of mutation and selection in the cultures. And in the paper, that is fully disclosed, that we think there was cross-contamination. But we also think that the results were showing, so just take it with a grain of salt. But like, we went through, I think, 3 rounds of evolution, and so we were saying, okay, probably in round 1 there was cross-contamination that caused this to happen. But the caveat to this is that there are some mutations that I am absolutely certain have been selected independently multiple times, and I hate them, and I don't know what to do with them. And we have looked at them, and we go and we use site-directed mutagenesis, and we put them in, and nothing happens. And then I tell people who do epigenetics about it, and they're like, oh, there's an epigenetic component. And I'm like, yeah, I don't do epigenetics, and I don't. But they're like, don't throw them away. I'm like, I want to autoclave them, and they're like, don't throw them away. But it's this thing where when you see mutations like that and you introduce them, they have no effect. And you can even, like, you can go and recreate the whole thing with site-directed mutagenesis, and you see no effect. But then in the culture, you see an effect. And people just say epigenetics. I don't know. Yeah. Yes. Yes, it can be real. It's just something that's really not reproducible with site-directed mutagenesis. I don't know. If you have ideas about it, I can send you strains. I would say I'd be happy to entertain them, but I've revisited this so many times, and I put so many students on it, and I'm like, I think I figured it out. We got it this time, and we go and do it, and it's the same thing. Nothing happens, and I'm just like, I'm done. We are done. So I can send them to you. Yeah, my X projects can become your problems. Okay. Yes?
So from your models, not only predict what mutants, but can you predict maybe how long it takes? You know, if you have 1 mutation, 2 mutation, how long it takes and how much antibiotics does it
Yeah.
take? You know, so what I'm getting at is basically, you know, if we want to control the antibiotic use, how much is really the limit that we can actually, you know, have the longest time of use for the mutation?
Yeah, those are good questions. I don't know the answers to those questions. Robert Beardmore is working on some of those questions. Like if we're cycling, so there are a bunch of people working on antibiotic cycling. Robert Beardmore knows all of them. If you want to get in touch with this community, contact Robert Beardmore, because he's like the social networker of the whole group. He's awesome. We're super friendly. He's in Exeter in the UK. And anyway, that's what he's working on, is how long things go. And he has all of these mathematical models. I'm not going to try to explain to you his mathematical models. Go read his papers and then talk to him about it. He's like a real deal mathematician. He does some experimental stuff, but he is like a true mathematician who has graced antibiotic resistance with his presence. And I mean that literally, not in a sarcastic way. Like, it's wonderful to have him in the community, giving proper mathematical treatment to this problem.
This question is going to seem like it's from left field.
Yeah.
But it still is interesting to me.
Sure.
All of what you're talking about is what I would call microevolution.
Yes.
There is also the whole subject of macroevolution.
Sure.
Like, you know, what determines evolution of body size,
Uh-huh.
or leaf thickness, or size of a seed.
Yes.
And I wonder if what you think, the things you have found, have to say about macroevolution.
Oh.
One thing comes to mind immediately, and that is, you say that some genes are have more evolutionary potential than others. I strongly suspect that things like, you know, major phenotypic traits like body size are influenced by a whole slug of different genes.
Yeah, sure.
So therefore, any one of these, mutations in any one of these can influence the ultimate phenotypic trait.
Sure.
So that's the one thing that emerges.
And okay, and so there are multiple genes involved in resistance, and they do interact with each other. These are the genes that have the largest effect, but even these genes don't have a fully predictive effect. So you're right, there can be multiple genes going on. Going back to the question of macroevolution and what this can say about macroevolution, I mean, this is just me being really wildly irresponsible, but I will tell you what I think. And that is, yes, and that is, and maybe, I mean, these are single-cell organisms, so there are limits to how macro we can get. But every antibiotic that has been discovered, tweaked, whatever, introduced into clinical usage is either directly from something that is produced by, it is either directly produced by some species of microbe, or it is a slight variant of something that is directly produced by some microbe. Right. And there are genes involved in these antibiotic production pathways that are shared across many, many species of bacteria. Now, there are few bacteria today that we know of that produce antibiotics. But then the other thing is all these resistance genes, I mean, they're ready to go. TEM1 has been found in cold seep sediments from the bottom of the ocean that are tens of thousands of years old. And it's just there. These are really ancient genes. And people are like, oh, they're co-opted from something else. I don't think so. I think it has been a beta-lactamase for 3 billion years. And I think beta-lactamases have been around for 3 billion years. So if you think back 3 billion years ago to what there was on the earth, there wasn't much. But there were bacteria, and they needed food. And so I think that they were using all of these things to kill each other and eat each other, which may sound like cannibalism, but they're less closely related than we are to cows and possibly to plants. So it's not, but I think they were just killing each other and eating each other. And I think that there's reasonable evidence for it. And so I think that that's what this says about it. And so as we're now bathing the entire world in antibiotics through agriculture, through hospital sewage, whatever, but mostly agriculture. It's just, there are these genes that have been silent, cryptic genes in the genomes of bacteria, and we're just waking them up. And then with just a single mutation, you can turn these things on. They are very easily mobilized to plasmids. Many of them exist in the chromosomes. TEM1 is on a plasmid, and it seems like it has always been on a plasmid. There does not seem to be an ancestor with a chromosome, but like CTXMs, those are coming straight out of the chromosome of Clivera. Now, it was dumped into Clivera by horizontal gene transfer from somewhere else, because they're not in the, Clivera is this group that has CTXM in it, and it's this unique Clivera thing. But you just pop it out of Clivera, stick it on a plasmid, and boom, it's taking over the whole world. And so, okay, and then so if you want to go really macro, there's this study I didn't show you. It was another phylogenetic study, but where I looked at how many plasmids there are. So I went back to like this old, what collection was it called? It was some pre-antibiotic era collection of bacteria, and looked at how many plasmids there were in those strains and how many plasmids there are in strains today. And back there it was like 1, maybe 0.5, not everybody has one, and if you do have them, most everybody who has them has only one, and then you get a few with 2 plasmids. Now, if you look at bacteria that we're collecting today, they have like 4 plasmids, average. That's just normal to have 4 plasmids. And so, I think in using all these antibiotics, not only are we causing these resistance genes to become active and to hop out of bacteria onto plasmids, but we're also causing bacteria to accumulate more plasmids, which is then increasing their ability to mobilize and disseminate resistance genes. And so I did this whole timeline study of it and looking at the emergence of new resistance genes, and it does indeed seem to be accelerating. Like, things are being mobilized a lot more frequently now than they were previously. So that's all I got for macro. That's as macro as it gets with me, but maybe it's something.
Thank you for this presentation.
You're welcome. Anybody else? We're like a few minutes over. I'm not offended if you guys are like, But I'm happy to stay and talk with you guys as long as you want to talk also. It's fun. You guys are, I really enjoyed you guys. Smart, fun group of people.